Hi, friends. It's Mr. Rogers here, and I want to talk really quick about phosphorylation cascade, which is one of the transduction techniques that we're going to be learning about in class. And so let me put up the notes real quick because you guys can see my webcam right now. So let me put it over here. There you go. So transduction is going to be, we got the message, we have the receptor, and we have those three different reception types. And now we actually have to move that signal from the cell membrane to where it needs to go inside the cell. Sometimes it's a nucleus, sometimes it's a different organelle. It just depends. And so there's different kinds of transduction, just like there's different kinds of receptions. Now, as it says here, reception sets off a relay team. Sometimes they're second messengers, almost like messengers outside the cell. Sometimes it's like a track meet where they're passing the baton to each other. Sometimes it's more random. Sometimes it's better. It's a lot like what we just went through with reception. There's different techniques, and there's different techniques for different situations. So almost all the time within transduction, I should say all the time, it's a multi-step pathway. And not only is it a multi-step pathway, transduction can amplify or increase the signal and create a larger response from just one single ligand. So one messenger could create 15 to 20 different reactions within inside of a cell going in different directions, which makes sense because, like, you know, if I'm like at a fire station and we get a call that there's a fire, not all of us are going to be driving the fire truck. Some of us are going to have to go do other jobs and get ready for it. Okay, so this requires a lot of coordination within the cell. This requires a lot of communication even within the cell. Obviously, we're talking about cells communicating with each other and how that goes about, but cells also have to communicate with itself, and it has to know what's going on everywhere within inside the cell. So there is multiple steps here, and obviously you can get really lost in the weeds with this kind of discussion, and you can sit here on cell communication for months and months and months, and might get into that when you guys get to college because this is a legitimate college course. It just depends where you go, right? So to add here, we're gonna only focus on one today, and that's because all the other ones are a little bit more. I just don't want to overload. I guess they're not harder; they just don't overload. So the one that we're going to talk about today is a protein phosphorylation cascade. So the protein, obviously, we're going to use a protein at the end. Phosphorylation, which means we're going to use ATP, and it's a cascade effect, meaning there's multiple steps in this process, and it's all moving down a path. Okay, so this is a series of proteins. We're going to call them kinases. They're going to add a phosphate to the next one in line, activating it and sending the signal to the target. So it's like a bucket brigade. So think about like if we're going to get a message and we're going to pass the bucket full of water to the fire to put it out or whatever. That's what a bucket brigade is. But I like to think about it as like a really messed up game of telephone inside of a cell. Okay. And then we're going to have to reset this cascade, and that takes some time as well. And so we'll discuss that because that's an interesting process in itself. Okay. So this is the picture, and you can pause. I'm going to have you pause right now. I want you to draw this picture. So pause. And welcome back. All right. So you pause, you drew this picture. Perfect. Awesome. Fantastic. Thank you for drawing an awesome, fantastic picture. I even forgot something in my picture, so let me add that real quick. So we're going to go through step by step, and we're going to do this twice. I'm going to do this once on the screen, and then I'm going to come back to my picture and go through it, and then add some notes next to your drawing. Okay, just like we did with the other ones. I should probably said this, like every page, we're going to have a different drawing. We're going to add some notes to it. Okay, so first thing, we have a receptor up here. It got a signal, and it's activated a relay molecule. So this could be a G protein. This could be a tyrosine kinase situation. This could be, this is not going to be an ion channel, to be honest with you. It's either G protein or tyrosine kinase, which tells you how this one is kind of viewed in a cell. it's not used for ion channels, so it's obviously going to be a little more important of a process. So we have a relay molecule. It's going to activate protein kinase 1. Now, this situation here is interesting because most of the time with the G protein, you're going to make GTP and whatnot. So this is going to require energy, but the energy is already produced by this receptor part up here. Okay. Or they could be the phosphates from the tyrosine kinase. So this guy is going to come down. He doesn't need an ATP to get him going, in the cell at least. And then he gets here. An ATP is going to come in with the active protein kinase, and they're going to activate protein kinase number 2. Okay, and you can see that this kinase has a phosphate on it. Once he is done, by the way, once he activates this guy, he gets reset automatically. He's going to go back and go be ready again. This guy, once he's come down here, he's going to meet up with the protein kinase number 3, the inactive version, with ATP again, and he's going to activate number 3. His process of actually getting back to inactive takes a little bit longer. We'll come back to that in a second. Okay. So you can see here, boop, boop, boop. It's kind of what I was talking about. Here, same idea. We have this activation of the ATP from number 2 with number 3. We have active protein kinase number 3. Now, I've heard of this system kinases going all the way to 9. I've heard it be 2. It depends on where the cell it's going and where the actual cell response is going to be, the active protein. Okay. So finally, this one is only 3. He's going to activate this inactive protein here with another ATP, and he's going to have phosphate on the end of him. And this guy is the one that's going to start the cell response. So this cell response right here gets started by this active protein. Again, he's got to get reset as well. We get a cell response. And that's what number 5 is talking about here. This takes time. So this whole process itself takes time, but resetting takes time. And this downtime means that we can't send multiple signals through the same cascade back to back. It takes time to reset. So you can't send one signal and then send another signal really fast. This is not a way for a cell to send back to back to back to back signals. It takes time to do this going down anyways. It takes time going back up. So that's important to know. So let's look at my picture here. So I have a picture, the same kind of idea. We have the ligand coming into the receptor. Therefore, he's sending a relay message. It's an active relay. A stands for active. IA stands for inactive. So he's going to activate kinase 1. Kinase 1 is going to activate kinase 2 with the help of ATP. Kinase 2 with the phosphorylation here, the phosphate from ATP, is going to be able to activate kinase 3 with an ATP. And you can see we have the inactive protein here. Same kind of thing we just went through. Straightforward. Okay, just my drawing version of it. So let's talk about some of the things that I kind of just brought up. So first thing I want to make sure that we're aware of is this takes time. It takes time. It also takes time to reset. So this is not something, like I said, you go back to back. It also takes time to go down the pathway. Another huge part of this is it requires energy. And using our critical thinking skills, when we just did the receptors, we were talking about, okay, if the receptor takes energy and it takes time, that means it's got to work every single time for it to be worth it. So this is a very successful technique. I don't know why I made that a T, I'm sorry. Successful technique. And it's something that is going to work almost every single time. Now, there are problems in the system. Like, this is for sure. This isn't like the receptors where tyrosine kinase has to work every single time and makes it perfect every single time. This guy does have a little bit more problems because it is multi-step. more steps, more problems. And you think about like anything in any solution in life, the more steps to your solution, probably the more problems that could occur. Same thing here. If I have 9 kinases, if every kinase isn't in the right area, if every little bit isn't in the right spot, it could go wrong. Now, why you do it this way? Because some of you guys are probably thinking like, Mr. Rogers, why don't we just have an active relay up here, turn on a kinase, and he just gets his butt down to this protein down here and activates him? Because that adds randomness. Small steps eliminate some issues of randomness. So if this kinase only has to travel this far, it's going to be more successful if the kinase has to go all the way down here, because what if the kinase goes this way and doesn't meet the active protein, or goes this way, or goes over this way and doesn't meet it in the right spot? Smaller, tiny little steps are easier for this process to work. So when I said there's like 9 steps, it's not always meaning that there's a huge gap between the things. There's always a big distance. Sometimes your cells have added more kinases to make sure that the steps are really, really, really small. They're incremental. They're tiny. So this process, okay, this phosphorylation protein, phosphorylation cascade is probably the most successful one we're going to talk about. However, it takes time. It takes time to reset. You got to spend energy on this. It's going to be successful, but there's a lot of steps, so there could be some problems. And there's small steps here because they're eliminating some issues of randomness. Now, this guy is going to be the one that is mostly associated with tyrosine kinases and G proteins, because they're already spending a lot of energy on those receptors. So we got to make sure that this process, it spends energy as well. It'd be pointless to use an ion channel here for this. So this is the first one we're going to learn. Make sure you write this down, and I'll talk to you guys later.